Hello and welcome to this episode of the SNEV podcast. My name is Will Phillips, and on this episode I'm joined by Professor Ivan Poon, a group leader from my home institute, the Trobe Institute for Molecular Science in Melbourne, Australia. Ivan's lab focuses on how and why dying cells undergo fragmentation into a type of large extracellular vesicle known as apoptotic bodies. I'm particularly excited to be talking to Ivan today as a small EV researcher myself, and we might not always consider the presence or role of apoptotic bodies and how they might contribute to EV heterogeneity. So Ivan, thank you so much for joining us.
Thank you. Lovely to be here.
So I thought I might start, this is a question we usually start off with to understand how people got into research and how their research journey started.
Yep. Mm-hmm.
So you did your PhD at the John Curtin Medical School at ANU, is that correct?
Yes, that's right. Yep.
Yeah. Did you start off working in apoptotic cell disassembly, or was that something you moved into?
No, I've actually moved into this quite late. I mean, when I was doing my PhD, it was all to do with how dead cells are being cleared. So I was already working on the dead cells area. But then after my PhD, then I did my postdoc, and my postdoc was something completely different. I was, again, going back to some of my biochemistry roots and, working on, how protein causes cells undergoing lysis, how the protein go oligomerization. It was actually my second postdoc that actually got me into the area by accident, actually.
Okay, yeah, cool. So kind of more of a protein biochemistry kind of background then.
Yeah, initially, but that was interesting. But what I did was, I was always interested in how dead cells are actually being cleared. And when after doing my first postdoc, I decided to go overseas to University of Virginia to do my second postdoc, again, trying to pursue this area of research. And I clearly remember, I always tell this story, I clearly remember my initial project was, to generate a particular transgenic mice. And obviously, while you're doing that, you do a bunch of various in vitro work. One day, I was just walking back from a flow cytometer, and my mentor just bumped into me and say, Ivan, one of the other postdocs decided to go into industry, and her project was working on how cells shrink when they die. So then I said, okay, as a overseas postdoc, I was happy to take on anything and give it a go. And then quickly work out within 2 weeks that the cells wasn't shrinking; they were actually undergoing fragmentation, and that leads to all the question related to how apoptotic bodies, and back in the days it wasn't considered as large EVs, how they have been formed.
Oh, very cool. So were you aware of what EVs were at the time, or was this something you just kind of followed and it kind of developed into looking at these?
Yeah, I mean, because exosomes and, I guess, the smaller EVs are well known back then. So we're talking about, in the 2011, 2012 time. So, and also, people I've worked with, they have, sporadically may have worked on some areas of, EVs, in particular exosomes. But I think it was then, at University of Virginia, there was only like 2 or 3 groups that were interested. And to be honest, actually 2 or 3 people, one of them is actually Uta. So we actually have a small interest group of 2-3 people to talk about EVs.
Yeah, it's sometimes you forget how small a world it really is once you get outside of the EV field. So you went to the University of Virginia, is that correct?
Yep.
And that was for a postdoc position?
Yeah, that was my second postdoc position there, yeah.
Okay, yeah, no, awesome. I mean, we had the MOVE consortium on a few episodes ago talking about going to a different lab, not just for obviously looking at new forms of research, but also for the cultural experience as well.
Yep, absolutely.
You've then come back to Australia and started working on apoptotic bodies, I suppose.
Mm-hmm.
So how did that research then develop?
I think at that stage, I kind of realized that yes, apoptotic bodies are recognized as a form of EVs many years ago. And I guess, but the problem was people always assumed that this process was random, not really highly regulated. But at the same time, that's when we discover this membrane channel called pannexin-1 was clearly regulating the process. And based off a lot of microscopy analysis that we can't see, no, this is something quite interesting. Cells are forming all these protrusions and so on. So therefore, I decided, and no one was working on this area, I guess, like what you're saying, a lot of people are working on the small EV area. So that I decided, I think there's a great opportunity to build a lab, in this area.
Yeah, no, very cool. So you've started looking at pannexin-1, you've looked at these disassembled apoptotic bodies. What were the things you started to notice? What are some of the intrinsic physical characteristics of these kinds of vesicles?
Yeah, well, I mean, the cool thing about them, easy thing about them is we can actually see them live being formed. So that has got, to think about, we can clearly visualize what some of the regulators are actually doing, like what I said before with pannexin-1. So for example, when this channel gets blocked, then the cells would then, more likely to form all this protrusion to help the cells to separate, the different blebs that actually happens during apoptosis. And I think, that actually got us all started, demonstrating that, it's definitely larger. There are definitely vesicles. And at that stage, because say back in 2012, a lot of the other large EV subset hasn't been described. So therefore, it's really the clear area of that to be explored.
Yeah, and I guess that's the interesting part about this kind of research is you can actually look down a microscope and see them, whereas with a lot of the small EVs, we have to go to an electron microscope or we have to use some kind of fluorescence tag to actually see.
Yeah, exactly. And then there's a lot of easy method that we can use by standard flow cytometry, rather than have to use more advanced flow that we usually have to do these days for the small EVs.
Yeah, something I found quite surprising when I was first reading about apoptotic bodies is there's actually a few different ways they can form. And one of the really cool things was the I hope I'm going to say this right. Apoptodia.
Yeah. Mm-hmm.
The beads on a string as they form off.
That's right. Yeah.
Could you tell us a bit about that?
Yeah, absolutely. So I guess the concept that we're trying to, the framework we're trying to establish is that every cell types, they may actually generate apoptotic bodies by slightly different mechanism. And it's actually a stepwise process. So what's well known is that when cells undergo apoptosis, a lot of the cells will actually undergo really, really dynamic blebbing, forming all these, circular bulge on the cells. And then eventually, some of the cells that do undergo fragmentation quite well will form this very thin string-like protrusion known as what you mentioned before, apoptopodia. Bits of death, that's what we call them. And then somehow, due to shear force or other, mechanistic force, they will actually then undergoing fragmentation. But then after looking at for a number of different cell types, we realized that, say, for example, a T cells may be able to undergo very dynamic blebbing and then form those protrusions, then form fragmentation of the cells, and then form quite large EVs. But then when we were working on monocytes, we realized that monocytes are not very good at forming those blebs. They're just some surface bleb. But they're very good at forming this protrusion. So therefore, that's when we discovered that when they do form the protrusion, because there isn't a bleb attached to it, the protrusion themselves actually form a beaded structure, like a beaded necklace. And that's the fragmentation of this that generates the large EVs. Because of those kind of properties, now we realize that depending on the cell, how well they bleb, how well they form the protrusion could lead to, for example, different types of, well, I wouldn't say subset at this stage, but apoptotic bodies or large EVs with slightly different properties and size.
Well, I suppose it's quite important because you would normally think, when you think of an apoptotic cell, you think of this big bulging thing with things coming off it. And you might think, hey, how is this different from the biogenesis of microvesicles or ectosomes? Whereas if you have this kind of beading coming off, it's actually quite a distinct biogenesis pathway then.
Yeah, that's right. Exactly.
So how tightly regulated is this process?
To be honest, we think it's very tightly regulated, and we are in the process of discovering more and more of the regulators. So, for example, we know when cells are undergoing blebbing, there are kinases like ROC1 that being cleaved by caspase activated to drive the blebbing process. And then the step 2, what we call it, is the protrusion apoptopodia forming phase. As we mentioned before, there's this channel called pannexin 1 that regulates that. This is a negative regulator. And then we have now found quite a few other channels and proteins that also can regulate this process, but we haven't solved everything yet. So basically now my lab is focusing on trying to identify all the regulators of this second step. So we think, based on our data, it's actually highly regulated process.
Yeah, and do you see much regulation differences between different cell types, neurons, muscle cells?
Very good question. We do see clear differences between immune subsets and as well as other like epithelial cells and so on. But in terms of really digging into the whole comparison between say muscles or neurons is something that we are trying to pursue, because I think one of the things that we can do obviously initially is let's look at all the different cells, give them an inducer and see how they form. And it's true, they do look quite different by microscopy, but we have also avoided trying to go for every single cell type. So we have described T cells, monocyte, endothelial cells, and there will be other papers that's coming through. But we really want to take this more to in vivo. So hopefully we can, actually be able to visualize how some of those cells undergo fragmentation in vivo, give it more biological context rather than just in the dish.
Yeah, because I imagine for example, between an immune cell and say just a regular other cell, we'll say. Mm-hmm. Yeah, you might expect to see quite some substantial differences.
Yeah, definitely. And also, I think even the same cell at different location, how they interact with neighboring cells, there's actually a lot to explore.
Tissue-derived apoptotic bodies, that's one I haven't thought of before.
Yeah.
So we have these apoptotic bodies. They come from pretty much they come from all cells, and it happens all the time. It's not due just to stress or things like that. It could be for a multitude of reasons. These EVs, we normally talk about apoptotic bodies in terms of large EVs, so kind of 300 nanometers plus, I would say, I think is what's usually said in the literature.
Yep.
Is it possible they could be smaller?
By definition, I don't think so, because I think obviously if you have a mixed sample, you can do different isolation methods, centrifugation method. You would definitely collect a variety of different EVs at different fraction. But in our area, we like to call the small apoptotic EVs. A lot of them are being called exosome-like, so they're definitely, some of the biogenesis do have comparable mechanism to exosomes and markers as well. But at the same time, for the large EVs, we argue that, yes, true, the definition of large EVs are bigger than 200, 300 nanometers, and true, we can't always see them by light microscopy. But because of what we argue that the apoptotic bodies are generated through a clear morphological change, and those morphological change can be captured by microscopy. So therefore, in large, we think that and then also, when you test all these different regulators, based on our experience, it's definitely, yes, there are sub-micron one. I'm sure there are apoptotic bodies that definitely delve into a range of maybe 800 to 700 nanometers, but I don't think they are as small as the smaller ones, yeah. But right now, the heterogeneity of this vesicle has to be examined first, until we know the regulators of some of those processes. Yeah, and we're still to be solved.
Yeah, no, absolutely. I think that's an important distinction to make, is we have something that we can actually see a biological pathway for. We don't need to, for an exosome and a microvesicle, we might not necessarily know which way it's come out because we can't see it. It's not very easy to track.
Yep.
So I think separating that kind of large apoptotic body from the rest is actually probably quite an important distinction. So yeah, we've talked a bit about size. We talked a bit about where they come from. We touched on cargo a little bit with Pennexin 1 and ROC1, but are there particular cargoes that you can then look for once you start looking for these particular EVs?
Yeah, what we do recommend though is that in terms of cargo, which also links to the characterization, is that for a long time ago, people always say, apoptotic bodies must contain histones because when cells die in fragments, the nuclear material often being distributed there. But based on some other published work from our group, and others as well, it's clear that nuclear content doesn't always get distributed to all of the apoptotic bodies. So therefore, nowadays we prefer to recommend looking for caspase cleaved protein, and that's often difficult as well because we also propose that we should probably look for active caspase free, for example, in the apoptotic bodies as another validation. But this doesn't always work perfectly because, depending on how much cytoplasm, the apoptotic bodies, when the morphological changes actually do occur, how much are actually being distributed, that may not always be the best. So from our own experience, going for a membrane protein would be better to highlight it is coming from apoptotic material because we have got, in our field of apoptosis, there are a number of membrane protein that are being cleaved, including the one that I mentioned before, Pennexin 1. So you can easily detect them by immunoblot as a good way to detect them. The other thing in terms of loading is that we haven't really, have identified in a particular protein or microRNA being distributed into apoptotic bodies. At this stage, it's probably more in the organelle level. So for example, if cells do undergo huge dynamic blabbing, nuclear material can be distributed into apoptotic bodies. But as mentioned before, there are cells like monocyte that doesn't go through this huge dynamic blabbing, and the apoptotic bodies generated from them actually contain really little nuclear material. So therefore, we think that, again, associated with the mechanism of how they've been formed, there can be material that cargo can be shuttle in there. But at this stage, it's not like we have got specifically partition certain protein in there at this stage.
It's something you touched on there as well, which I think might be interesting to talk about more. Is you have these apoptotic bodies; they've been selectively produced by the cell. Why?
Yeah. I mean, this is a very good question, and I mean, people have multiple different approaches to do this. So one argument is when a cells fragment, because of the idea of, because at the end of the day, the whole concept is when cells die, they have to be cleared away. So therefore, when cells undergo fragmentation, it's analogous to, for example, you have a big piece of steak, you can, you can't just shove it, or some cells can't actually eat the big thing, right? So therefore, for it to undergoing fragmentation to smaller pieces, make it easier, just make it easier meal to eat. But at the same time, there are the concept of, for example, because it came from a cell to begin with, it will have a variety of different cell surface receptors and so on, or proteins or signaling molecules. So therefore, if those are still on the apoptotic vesicles, they obviously can perform their particular function, assuming the apoptotic bodies are not being cleared. And alternatively, we also think that, say, viruses can definitely hijack the process by hiding within the vesicle or on the vesicle as a way to deliver to the payload to the another cell. So that's kind of the function that we are getting at. But I'm hoping a lot of those experiments at this stage are still done in a situation where we basically isolate the vesicle, and then either treating other cells with them, or administrating back to the mice or whatever. But what we do hope is that when we do know the mechanism of formation, it's always better to approach it that way with other genetic approach to see whether modulating regulators that control specifically the formation of apoptotic bodies.
I find that interesting. I like the steak analogy, by the way.
Yeah.
It's very good. So I suppose it would be macrophages then coming along and taking these little pieces of steak or these apoptotic bodies. Do you know how they might be targeted, or say, hey, come and eat me like I'm an apoptotic body?
Yeah, definitely. So in the field of cell clearance, it's very well known that, for example, their metabolite that can be released through the dying cells, and also in our case, apoptotic bodies will continue to release some of those metabolite to attract the phagocyte. There are also work that's been done showing that certain chemokine can be located on the surface of apoptotic bodies, and that could attract certain phagocytes such as macrophage. But at the same time, when cells die, because as you mentioned before, cells can die anywhere. So we're still to define what kind of different types of phagocyte that could interact with apoptotic bodies, and depending on the pairing of what is dying, what is there, the downstream response will be quite different.
Yeah, and I imagine you wouldn't want all of your cell contents spilling out into the extracellular space, as that might cause some kind of severe immune reaction, and you might get all kinds of unwanted effects.
Yeah, absolutely. And we actually just recently published a work on how the apoptotic body. So one of the concept of is actually EVs in terms of the stability of the EVs being controlled. And obviously, a lot of the work has been done on a small EV area where they're extremely stable. You can freeze them, you can do all these things with them they don't like. But we actually discovered that our apoptotic bodies, how stable they are, is actually controlled by a protein called NINJ1, which has been discovered in the field as a key regulator of membrane lysis. And we also find that to be quite important in terms of after the cells has undergone apoptosis, after they have actually generated the apoptotic bodies, a particular protein, when they oligomerize, regulate the vesicle stability.
That's very cool, and something I've always been a bit interested in as well is this idea of once you have an EV population outside of a cell, there might be enzymes or other factors there that mean that population is still active, it's still doing things. So populations can change. So apoptotic bodies don't survive very long outside the cell then?
Yeah, I mean, from our experience, at least under in vitro setting, they will probably undergo lysis within 4 to 6 hours. So therefore, if you have to work on apoptotic bodies, I think this is something to keep in mind of, is that you do have to work fairly quickly with what you have to do a function assay, and keep in mind that, yes, for example, you may induce cell death for 24 hours and then collect stuff, and then then do your next experiment, wait for another 48 hours, whatever, right? But then we always keep in mind of, for example, if cell death are induced asynchronously, some apoptotic bodies would be lyzed by the time you collect them. So, and then also if you throw them onto assay and wait for 24 hours, you are looking at the effect of the initial stage of interaction as well as when the things lyse. So all these things have to be taken into consideration when you think more deeply about how they're being generated.
I think there's some implications there as well for any kind of EV research, because you've got your sort of self-culture media sitting there with stuff in it floating around. It might be there for, you might do a 24 or 48 hour incubation. Those apoptotic bodies, they might not completely disassemble into strips of membrane. They might reform and form smaller particles.
Exactly.
Definitely something to think about. Moving on, we've got all these markers and things, and we know about apoptotic bodies. How do we isolate them?
That is a easy and hard thing to do, I think, is that because they're large, so it's quite easy to separate them from the small EVs. But the problem is, because they're also rather large, the main issue that we often face with is, are we separate them again away from apoptotic cells that hasn't fragmented, depending on the cell type, whether we're affecting viable cells, and more problematically is also often necrotic cells when they don't pellet really well. So we do use a differential centrifugation method to do so. whereby usually the small EV people will spin at 500 Gs or whatever 300 Gs to remove the cells, and then may do further spinning to remove the large mid-size range EVs, and then do red high spin to do the small EVs, and we basically capture them early on. So for example, we will try to remove cells from 300G, and then at that stage we do another 3000G spin to try to isolate apoptotic bodies, and usually from my experience it's pretty good, but again it depends on how your cells are being cultured, the cell types and so on. Sometimes we do have to tweak that a little bit to ensure we get a more pure population, but that's more of the crude way of doing it, for example, to do initial characterization of mechanism characterization or doing certain functional studies. But when we have to do functional studies that will have to be squeaky clean, we do move to the fact sorting, because as I mentioned before, they are large enough to be sorted on a standard fact sorter. So that's one of the methods that we've come up with to sort this kind of large EVs and allow us to put markers on them, like do they expose phosphorylserine as a marker to actually isolate them. But unfortunately, as we discussed before, the stability is an issue. So again, we need to be mindful of how long we are using the sorter to isolate them before we do any functional or downstream analysis.
Yeah, you can't just stick it on the bench for 20 minutes and walk away. It sounds like you have to be pretty quick. So yeah, obviously with isolation, and you did touch on it a bit, something as a small EV researcher I'm always thinking about, particularly working with biofluid, is the presence of non-vesicular extracellular particles and other contaminating particles. Is this something you guys think about a lot?
Yeah, we do. I think what we haven't done too much of is using the differential centrifugation approach to isolate apoptotic bodies from in vivo setting. But that's why when we do flow cytometry, that's when we can add certain markers in there to isolate from there. And I think it's still a long way to go. I think with the large EV isolation, I think there's a lot of opportunity to develop clever approach. We have thought about microfluidics, we've thought about affinity capture and a few other things to play around to develop better ways to capture those EV subsets.
Yeah, definitely. And the plethora of tools we have for small EVs, you would hope that eventually some would come along for the larger EVs. Yeah. I mean, is there anything much more you would want to add onto that before we move on next?
No, that's but I mean, we can often talk about, I think the major thing that I would like to highlight is, and what we're trying to do is, how can we distinguish apoptotic bodies from other large EV subsets? Yeah. I think apoptotic bodies has been recognized as one of the earliest described large EV, but it's pretty clear in the last 5 to 10 years since we first described some of the work, there's just so many other large EV subsets that completely different to apoptotic bodies, like microsome, exopher, land vesicles, and large oncosomes. There are many more, right? So I think I hope to work with a lot of the other large EV experts to really tease out some of the different mechanisms, different markers on them to really benefit the field here.
Yeah, 100%. And it's almost every day someone's got something new, or you go to ICEV and there's a new large or small EV type that somebody's talking about.
Yep.
So yeah, definitely a lot going on. So what are kind of the main challenges for apoptotic bodies at the moment? Is it method limitations like we talked about a bit before, or is it more in the mechanism and disease implications kind of route?
Yeah, definitely. As I alluded before, there's still a lot of methodology development of understanding what are the markers on them that can clearly distinguish them from other large EV is definitely a problem. But in terms of mechanism, something that in vitro wise, I think we are getting there. I think in the next few years, we will definitely have a much better understanding of how large EVs through those morphological changes are being formed, and a lot of them can also be confirmed by in vivo. But I felt like the key thing to do next stage is definitely in vivo mechanistic studies, because we have done a lot of work that hopefully will be published soon. That is, the neighboring cells plays a key role in driving that process. But I guess demonstrating they have a function is probably one of my key goals in the next few years. It's because, yes, there are people that demonstrate that, including ourselves, fragmenting them could make them easily cleared. They do harbor in certain signaling molecules, but trying to demonstrate that they actually play, when you manipulate them in disease setting and actually and change the progression is what we really need to move forward to, because that will lead us to connect a mechanistic understanding to potentially a therapeutic outcome to manipulate how they're being formed.
Definitely. I mean, is there diseases people are looking at in particular for the role of apoptotic bodies at the moment?
Yeah, a number of groups have worked on the virus transfer, including ourselves. So that's probably what we will be pursuing, putting a lot of effort in, as well as expanding onto other way the autoimmune inflammatory disease is again of interest of people as well.
Yeah, the autoimmune or inflammatory disease angle would be quite an interesting one. Do you think there's space for perhaps small EV researchers to come in and help with some of these research questions in the apoptotic body field? Or perhaps there's other fields we might not even think about that you're thinking about that might be useful for your field?
Yeah, absolutely. I think the small EV fields, because of how you guys think, how you characterize, and then also some of the study definitely can be applied. At the same time, unfortunately, there's a lot of things can't be applied because we're working on two different things. I felt like a lot of the methodology would be different, but at the same time, there could be a lot of crossovers, and we definitely learn a lot from the small EV fields of how, for example, for engineering point of view, detection point of view, characterization point of view, we will still follow those kind of guidelines. At the end of the day, is EV formation. But at the same time, we take a lot of inspiration from the cell clearance field because at the end of the day, biologically, because apoptotic bodies are by definition formed exclusively during apoptosis, and apoptotic cells must be cleared in some way. So therefore, there is a lot of crossovers between the two different fields in terms of what are the biological function of apoptotic EVs. And then we also have this association with the cell death field as well, because we know that people are always interested in what kind of proteins have been activated during apoptosis. Like what I mentioned before, knowing that people were studying Ninja1 as an important membrane lysis regulators, now we realize that it also plays a key role in the vesicle lysis. So we do have to learn from the EV field, from the cell death field, and from the cell clearance field, and using those knowledge together that sit between all those areas.
No, absolutely. And I think it's so important as well to go and look at other fields and think about what are some things I can apply here, or even there's might be knowledge that you can offer. Definitely, that's something I've experienced with some of my collaborations where I work with people who aren't EV scientists and having to explain the methodology and why we do things. But at the same time, I still get quite a lot of useful knowledge from them as well.
Yeah, absolutely. I think another thing that is also cross-disciplinary, right? So I think in something that we really want to work on, but we haven't, I guess, put enough resource on is the biophysical properties, because we know from our experience as well that a lot of the process that we see may not necessarily be always driven by like a protein oligomerization, assembly mechanism, whatever, or signaling mechanism. It could simply be just in the biophysical. So therefore, if anyone's working in this area listening to this, happy to have a chat and, see whether there are, good collaboration that we can have.
Oh, absolutely. And I think that's going to be quite a hot area for us over the next few years, because we have mRNA therapies coming out and these engineered nanoparticles. I think there's much to be learned about native EVs and how they can be applied to that field, as well as how we might be able to take some of their tools and apply it to what we do.
Yeah, absolutely.
We're coming close to the end of our time together.
Yep.
I thought we might talk for a bit about, do you have any advice for younger researchers? Like what are some of the things and skills that you think might be important now for young researchers to develop?
Yes. I mean, I think for young researchers, it's very clear, obviously your own expertise, do things carefully, do things well, expand your knowledge of multiple different fields to digging very deep into the mechanistic understanding and so on. I think those are very obvious. And then, I'm sure people have talked about all the networking kind of related things. But something that's very close to my heart is actually more to do with expanding on trying to learn how to recognize an area that people haven't explored too much. Don't want to just make small modification, keep going. Sometimes that's important. I definitely agree, making some of those steps is critical for the field. But at the same time, for my own path, I often find that finding areas where people are not really working on at that stage and really interest you is probably the most important, because I find that create a kind of niche area for yourself to expand in, to be differential to other people. I mean, for example, I would have recognized that many people was working on the small EV field. So therefore, I decided early on, I will work on this large EV area that no one is actually working on, and it aligns with my own interests. And that will really help you to have papers that will have that kind of novelty to explain to people why this is different to other people, as well as helping you to progress to the next stage where you can establish your own research program that is very different to your mentor. And to me, it's just fun, because I think we have to talk about a lot of unknown. I probably tell you that we don't know more than what we know. So I think that's the fun part, because there's just so many areas to work on, and there's not a huge competition at this stage. Everyone can work well together. So that's what I like.
Completely agree as well. I think it's so important to kind of try and find something that you can build your niche in, or build into a niche. As well as I think you need to kind of just ask questions, and don't be afraid to ask dumb questions. I ask dumb questions all the time.
Assumption as well. Don't always make
Yeah.
assumptions, because I think from a body's large EV point of view is that people make a lot of assumptions initially about how process being work. And I think, until you see the data, that's the key thing. Don't just think oh no, this shouldn't work this way or that way. Because I felt like the EV field has other field have often criticized us of they never release enough EVs to do so. What's the meaning of isolating all these EVs and then inject them back to mice? Until you actually in it, you often make a lot of assumption about how things work or don't work.
I think as well as when you do get those kinds of criticisms, I think it's just also important to acknowledge that that there might be a little bit of truth in there as well.
Oh, absolutely.
And you need to be open to other possibilities. And what you said before is like, we don't know everything. We deal in uncertainty. So it's so important to just always be mindful that what you're doing, hey, it might not work out, but that's the nature of the game.
Exactly.
All right. Well, I think that about wraps us up, Ivan, unless you had anything else you wanted to sperook or talk about before we finish.
No, all good. Oh, good. Thanks for inviting me to come along.
No, thank you so much. And it's been really great. We're hoping to do a few more of these where we can have perhaps some non-small EV people come and talk about their particular vesicle of interest.
Good.
Excellent. Thank you so much.